Hello, everybody. Welcome to the second Revive seminar. I am François Franceschi, project leader of the Antimicrobial Memory Recovery and Exploratory Program, AMREP, at the Global Antibiotic Research and Development Partnership, GARDP, in Geneva. Revive is part of GARDP AMREP program. It is an online space to connect and support the antimicrobial research and development community. It aims to facilitate learning, to connect people, and to share knowledge. This webinar series is part of Revive's educational activities. In addition to our online webinars, we also co-organize sessions at key conferences in the field. As Revive aims to support the antimicrobial research and development community worldwide, we organize follow-up webinars to make our conference sessions available to a wider audience and also to give people the chance to ask questions. This presentation was part of the session Expediting Antibacterial Development, Core Lessons and Key Tools for a Rocky Road at the European Congress of Clinical Microbiology and Infectious Diseases, ECCMID 2018 in Madrid last April. This session was co-organized by GARDP and CARB-X. Today's presentation will be followed by a question and answer session. Please use the question pane on your webinar control panel to submit your questions. You can submit questions at any time during the presentation or once it is finished, but please note that the questions will only be discussed after the presentation. We also will do our best to respond to as many questions as possible in the time we have allocated. I'm happy to welcome today's speaker, William Hope, from the Center of Antimicrobial Pharmacodynamics at the University of Liverpool, as well as our moderator, Ursula Theuretzbacher, from the Center of Anti-Infective Agents in Vienna. And with this, I would like to hand over to you, Ursula.
Thank you, François. I'm Ursula Theuretzbacher, microbiologist and expert for antibiotic R&D, especially interested in pharmacokinetics and pharmacodynamics. I'm delighted to moderate today's session and to introduce our webinar speaker, William Hope. He is Professor of Therapeutics and Infectious Diseases at the University of Liverpool in the UK. Since 2017, William leads the newly formed Center for Antimicrobial Pharmacodynamics at the University of Liverpool, which provides the pharmacodynamic packages for the development of new antibiotics. William is well recognized for his research on pharmacokinetics and pharmacodynamics and supporting antimicrobial drug development and individualization of antimicrobial therapy with PKPD strategies. Welcome, William. Please start your presentation.
So thank you, Ursula, and good afternoon or good morning to everybody. So here's the first slide. This is a question that is increasingly being asked by a whole variety of investigators and drug developers, given the importance of PK and PD. And in a sense, it's a rhetorical question that scientists often face. So how many technical replicates are appropriate? How many experiments should be done? And how do you know when an experiment that you've done is right? And George Strasano, when I was his fellow, taught me that one of the answers to these questions until you're sure. And then, of course, that begs the question, what do you mean by sure? And I've come to realize recently that enough PK and PD is being done when you're confident to be the first person to administer a new drug to the very first patient. So the role of PK and PD in drug development is recently become increasingly important because it provides the supportive evidence for causality, and that is that the benefits of a drug are a result of the observed effects from the drug itself, and that it exerts its effects by predictable biological effects that can be harnessed for therapeutic benefit. And these ideas were first developed by Dr. Peck when considering the necessity for more than one clinical trial for drug registration. And in the days that Dr. Peck was writing, the FDA would require 2 trials to ensure that a drug was really exerting an effect and that it wasn't being effective by chance. But more recently, we've seen that PKPD is an alternative way to establish causality, and as opposed to conducting multiple comparative clinical trials. So the importance of PK and PD has been recognized by both European and American regulatory authorities. The EMA has gone as far to codify what they're expecting to see from drug developers in development packages. And this slide shows that document, and I'm going to read out some of the essential parts of this document, and these are excerpts. So for reasons of lack of feasibility, that is of clinical studies, and as part of abbreviated clinical development programs that address unmet medical need, it is essential that there are very robust PK and PD analyses to support the likely adequacy of regimens. So that sentence replaces, there's no excuse or requirement to guess the dose. It must be supported by a robust PK and PD. There's a sentence that refers to the use of PK and PD to minimize or replace dose-finding studies that were characteristically done in phase 2. PK and PD has a central role in regimen selection, a selection of regimens for special populations, such as critically ill patients or children or neonates, and also goes on to talk about selection of regimens for the minimization for selection of resistance. So this document also says in towards the end of it that it's not a detailed guidance on the methodology that's used. So it doesn't tell people how to do their lab animal models or their hollow fiber models, or indeed how to analyze their data. It does allow for alternative strategies, and this may be increasingly important for the development of adjuncts to antibiotics, which is an area of rapid or increased interest. But it does suggest that there needs to be early discussion with the competent authorities if that's being pursued. And it also acknowledges that the PK and PD field is mature and it's fit for purpose, but it itself is developing quickly. So I want to just make the point that there is no PK and PD recipe book, there's no grand plan, there's no roadmap, there's no text or there's no mantra. There's no way that you can write out very specifically from the start what a PK and PD program would look like. Our own experiences, our plans generally change very quickly from how you write them down as the first data comes back. And I don't think that there's a right way to construct a package. There are multiple different ways to construct a package that would be fit for purpose for drug registration. For sure, programs require very good communication, and they require a lot of trust, and they require higher degrees of cooperative working between sponsors and pharmacodynamic laboratories. And if those relationships aren't very functional, then it's very difficult to get a good package. For sure, PK and PD is not cheap. Although arguably it's much cheaper than doing phase 2 clinical studies, and it's certainly much cheaper than having failed clinical programs. So while it's not cheap, it generally is fun because of the learning that happens and the collaborations that develop. I also just want to make the point that it's very dangerous to make too many assumptions about the pharmacodynamics of a drug. Even a drug of an established class, each drug is different in the way that it behaves, and it's I think it's much better to do the experiment and interpret the experiment rather than design the experiment expecting a result that somehow you believe should be the truth. So do the experiment, be guided by the data, and to enable the planning for the next experiment. So the first big task for any PK and PD package, and this step happens typically quite early in the program, is the determination of the relevant pharmacodynamic index. That is the link between the dose of the drug, the PD index, and the outcome of interest. And so typically there are 3 measures, or 3 PK and PD indices, and here are the classical ones. And here I'm showing a dose fractionation experiment that was performed, I think, originally in Bill Craig's laboratory. And on the y-axis here, the bacterial densities in a mouse. And here are the 3 measures on the far left. In A, there is the peak to MIC ratio. In the middle is the AUC to MIC ratio. And on the right is the time above threshold, which is typically the MIC. Typically these experiments are done with finding out with nonlinear regression which curve best fits the data, and you can see here clearly on the right the time above threshold does the best job of explaining the experimental data, and these results would be typical for beta-lactams such as penicillin, and these terms are very well embedded, I think, in the general literature, but also in regulatory and drug development science. So I've come to learn that dose fractionation studies look very pretty when they're presented like they are on the previous slide, but actually in reality they're very hard to do and hard to do well. And they're hard to do for a number of reasons. They're generally very large studies, and they require a lot of knowledge to be able to execute well. But they require, and they serve 2 major purposes. And the first is a general narrative that's expected by many, that is the regulators, for example, and investors and other scientists wanting to know whether a drug is Cmax to MIC, AUC to MIC, or time above MIC. And at the recent ASM microbe meeting at Atlanta, that question was repeatedly asked in the new agents session at the start of the meeting. But it's clear for those of us that do these experiments a lot that very complex biology is actually shoehorned into one of these 3 measures, even if it doesn't fit well. So for new drugs and new organisms or new targets, there's a lot of pressure to try and just make very complex systems fit one of these 3 measures. And sometimes that can be difficult and throws some challenges. And it's the nuances that these studies throw up that are important for pharmacodynamicists, and this is where there's a whole range of things that are important in understanding drug effects become apparent. So hysteresis is a mismatch between serum concentrations and tissue concentrations. Effect site concentrations become important. Drug receptor interactions, protein binding effects, immunological effects, emergence of resistance clones, tissue-specific effects, driver switching, so time above MIC in one model system, but AUC in another model system, for example. And all of these things can come together to complicate the story rather than the very simple case that I showed in the previous slide. And understanding why that's the case and being able to explain it to a whole range of people, so sponsors to regulators to academics to funders, becomes very important. So almost all of preclinical PK and PD is done in experimental pharmacological models, and really there are two sets of main model systems. The first is the laboratory animal models, and that is predominantly mice. And mice have the advantage over other systems of mimicking biological barriers. I mean, for example, they have lungs and brains. They have immunological effectors, and even neutropenic mice actually have immunological effectors that can confound the effect. One of the advantages of mice is that these models are not confounded by resistance, because resistance doesn't have time to emerge, and the inocular that are used in these model systems is not sufficiently high to enable resistance to rapidly emerge. You can mimic or assess the importance of effect site PK. There are two common models. The first is the thigh and the second is the lung. There tends to be less variance with the thigh, but there tends to be more effect that's observed with the lung model. And then on the right here, there are a variety of in vitro models, so pot models and hollow fiber models. And the advantage of using these models is that there's an ability to examine the pharmacodynamics of resistance. There's an ability to escape from the limitations of laboratory animal PK. And there's an ability to more easily perturb the regimen to uncover relevant biology. So you can use very high regimens or unusual regimens that would be very difficult to do in a mouse. For example, infusing drug. But I think that there's a preconception which is wrong, that in vitro models are somehow easier and that they're somehow cheaper and that they're somehow faster, and none of that's true. Unfortunately, these tend to be more complex models and certainly more expensive models than murine models. So the difficulty with dose fractionation studies are that it's uncommon to get the experiments properly centered the first time. So that means that there has to be a lot of pre-existing knowledge about where to set these experiments up so that the relevant biology can be demonstrated. And in our own experience, we generally don't get this right in the first few experiments. It takes a while before you understand the drug and the disease well enough to get the experimental conditions right. The next important problem is that some of the effects are relatively small. So in these experiments, variance is the enemy, and you have to set the model up so that you control variance very tightly, so you can distinguish real biology from noise. And then there's also, it's important to have a very good understanding of PK and PD and the design principles, so that the schedules that crowd too closely around the half-life in a mouse, everything looks like an AUC drug. Even a penicillin might look like an AUC drug in that case. If schedules stretch too far beyond the half-life, then everything looks like it's a time drug. And it's important, again, about this centering question, that if the experiments are fractionated where there's no effect or there's maximal effect, then everything must return as an AUC. So you can't determine whether things might be time dependent. So the next big question after the dose fractionation studies have been conducted and there's some idea about the relevant index, is to come up with a pharmacodynamic target. And that is the magnitude of effect that is likely to be clinically relevant. And it is conventional to quantify this in terms of stasis, so that there's no net change in the bacterial density following the initiation of therapy, or various orders of logarithmic killing, so 1 logs, 2 logs, and so on. The triple log refers to the idea that there are 3 moving parts and that they all interlock, and if you change one, you'll change the other. So down on the bottom left, there is the idea of covering the wild type. That's being able to treat organisms without intrinsic resistance mechanisms. This requires a very well-established clinical microbiology program that enables a definition of the wild type to be made. And then down there in the bottom right, then there's, at this stage, of the program, there's generally some phase 1 data and maybe even some phase 2 data if those studies have been done. So there's some idea about what's possible to be achieved in the clinic, and also whether the exposures that are being experienced by humans are within safety margins and whether they're well tolerated. So these three things will move together, and if one changes, like I say or said, then the other will change, and that's what the next few slides are going to show. So just to talk about the definitions of stasis and 1 log kill. So this yellow dot shows that mice that were infected with Staph aureus, this is MRSA, achieved at the time of initiation of therapy 6 logs, and then the organism expanded out in the course of the experiment and grew 2 logs to achieve at the end of the experiment 8 logs. And then this drug is teicoplanin, and various measures of drug exposure are shown on the x-axis, and this drug's been quantified in terms of the AUC to MIC ratio. And the red dot here shows the amount of drug, which is approximately 600, that's required to achieve a 2 log kill. And you can see there are also the other measures or estimates for a 1 log and a static endpoint as well. So what's required to accurately determine the magnitude of a pharmacodynamic index that's required or relevant for the patients? And so here, this is more conventional than having a large amount of precise data, but I think many of us say that us using or taking a static endpoints relevant for a less severe clinical indication. And so by that, we generally mean complicated urinary tract infections. But for more severe diseases like pneumonia, orders of logarithmic killing are generally perceived to be important. It's also important in terms of setting the pharmacodynamic index or establishing the magnitude that's relevant, that a wide number of organisms or relevant organisms are studied. And so that means studying different strains, potentially different species and different genre that are expected in the clinical program. It's not relevant or possible to study all of them, of course, but and that's part of the topic for today's talk, but a reasonable number, so certainly more than 1 strain. And so we're going to get on and talk about how many more than 1 is relevant. So for gram-negative programs, for example, then, and certainly for urinary tract, complicated urinary tract programs, covering the leading pathogens is clearly important. So that might mean covering E. coli and Klebsiella pneumoniae, but it may not be feasible or reasonable to cover every member of Enterobacteriaceae. And then how many strains of each species? Well, our own practice has been between 4 and 10, and of course, again, until you're sure, and you have to let the data guide that decision, but it's not 1 and it's not 2 and it's not 3. But after a suitably large number of strains, the overall pattern should become apparent. And it's important to include in that panel resistance mechanisms, which is often the topic of unmet medical need, and it's important to demonstrate that, and that's the topic for the next few slides that are coming. So I'm showing here on this slide, these are made up data, but they're also based on real data that we have seen. And so this drug is a time above MIC drug, and you can see on the x-axis there are the scale going from 0 up to 1, so from no time above MIC to 100% time above MIC, and on the y-axis there is the number of strains. So here we've taken a strain, and for each strain we've estimated how much drug exposure is required to achieve a static endpoint for that strain. And so here there are 12 strains. And the point that I just want to make here is that this curve is not normally distributed. It's log normally distributed. So the median is different from the mean, and it's certainly different from having an endpoint that would take everything. So there's a large difference you can see between having a median value, which might be 25% above time above MIC, versus the mean, which might be something like 40% time above MIC, versus having to look after everything, which would be 90% time above MIC. And quite how this problem gets resolved is there are no rules about how this is resolved, although most investigators would take some measure of central tendency when trying to set a pharmacodynamic index that's relevant. So I spoke about the importance of taking strains in or studying strains with different resistance mechanisms, and there are two key ideas here. The first is the providing evidence that the MIC is biologically relevant. That is, it's transmitting biologically relevant information. And so that means really taking strains with MICs that are within the wild type and those that are beyond the wild type or outside the wild type. And that if you can show that the MIC controls the pharmacodynamics, that is that so that a strain with double the MIC requires double the drug exposure to achieve the same effect, that's very powerful evidence that the MIC is likely to be helpful in the development program and relevant for patient care. But there's also another reason to take strains with different resistance mechanisms, and that is this idea that that demonstrating the lack of cross-resistance of the new drug that you're trying to develop. So if a drug is completely naive to the presence of another resistance mechanism, then the pharmacodynamics of the new drug should be exactly the same as the wild type. And this is an important point that is actually in the EMA document about the explicit demonstration of the lack of cross-resistance is a very important principle. So two separate but interrelated ideas for trying to use strains with different MICs and resistance mechanisms when you're doing the PD studies. So I referred a few slides ago to the triple lock, and this is how these data are brought together. And I will just show you the way that these things can move together, and they're interdependent. So this is a common plot that's used by pharmacodynamicists and drug developers. On the x-axis is the MIC. On the y-axis this is usually the probability of success, and it's usually the number of simulated patients that achieve an endpoint that's deemed to be important. So here, it's an AUC to MIC target of 88.8. So what happens is that in a simulator, there's a calculation that's done about how many simulated patients achieve that pharmacodynamic target for each MIC value. And you can see that for low MICs, 100% of patients manage to achieve that target, but as the MIC climbs, then patients start failing. That is, some of the simulated patients don't achieve the target that's deemed to be related to success. And also, it's typical to plot the MIC distribution on this same plot, and you can see that the epidemiological cutoff value for this particular organism is 4 milligrams per liter, and that these candidate regimens that have been proposed on the basis of phase 1 data generally would all be successful in the sense that they would cover the wild type satisfactorily. And so I'll just make the additional point that these calculations in this example were done by using stasis as an endpoint. So this might be relevant, say, for example, for a less severe infection like complicated urinary tract infection. But if you require a more rigorous target, such as for pneumonia, if we go to the next slide, you can see now that there are many fewer patients that manage to get the target. It's much higher now at 610.4. And that's moved now because more drug is required to achieve a 2-log drop in burden. And you can see that most of these regimens now sort of bisect the wild type, and the prediction would be that this drug wouldn't be a safe or these regimens wouldn't be safe to develop in clinical programs, that their risk would be too high because too many patients would have concentration-dependent clinical failures. And so this tells you that this would not be potentially a safe option to pursue in subsequent clinical studies. So I think it's also important that it's very easy to get sanctimonious, if I could use that word, or get too rigorous or get too greedy in drug development programs. And so that it's simply that you can't have your cake and eat it, as the UK's Foreign Secretary says. So you can't have a very rigorous endpoint such as orders of logarithmic killing and suppression of resistance, and then cover every strain that's out there in the world, which have some of which have very high pharmacodynamic indices, and then expect to have very high target attainment rates right across that distribution of MICs. It's simply not possible, and if you demand that, then you'll be disappointed, and the drug will not have the you won't be able to establish a PD relationship that's enables subsequent development plans. So there has to be some degree of compromise so that you don't needlessly kill a drug that's otherwise perfectly good. I think that the important point here is not to rely on absolute numbers, but to use well-established positive controls, use drugs for which licenses are established and for which there's a large amount of clinical experience, and benchmark against that drug, so that at least enables you to show that your new drug is performing in a comparable way to what might otherwise be accepted and established. And this comes to this idea of benchmarking. So if you show a relationship like this to a medical student and you ask them to take the endpoint that they would like to bring into a patient study, you can see that that is generally, they'll point to where there's maximal activity, and that's here, this circle. But a benchmarked endpoint may be where a suitable positive control or comparator performs in the model. So remember, these models are not absolute, their performance reflects the way the experiment is designed. And so the importance of using a benchmarked endpoint is to at least match that, and certainly not design regimens of the new drug that goes to the left of that benchmarked endpoint. So I think benchmarking is incredibly important, so that you can just, rather than dealing in absolutes like taking too much effect, which just may not be possible, is that you just try and at least benchmark or match what the comparator is. So this is the back of my good friend Tom Parr, who's the Chief Scientific Officer at Spero Therapeutics, and this is a real email that he wrote me a few months ago, just before I wrote this talk, and I thought that this was exactly what I wanted to say. So that if we can tell about the pharmacodynamic index, we will get the dose and all will go forward. So I think that that is true. But if we can tell the story in more than one way, and it seems to hang together by, so to speak, triangulation of the stories, it is even better and more likely correct and useful. So Tom is talking about different perspectives to make sure that conclusions that are coming out of these studies are rigorous and fit for purpose. And so in the next slide, I have started writing some of these ideas down. So this concept was actually first brought to my attention by John Rex, who calls this orthogonal reasoning, so getting to the same answer via different pathways. Alan Forrest taught me about exercising or stressing model systems, so not using the same sanitized model over and over again, but using different model systems and making sure you get the same answer. For sure, using more than one model system, so another laboratory animal model, hollow fiber model, and of course there's the problem or the risk if you use different models that you can get discordant results. They teach you different things, and it's important to actively manage and seek explanation for that discordance. It may be that you decide to use more than one PD lab, It may be that you use more than one study endpoint or readout. So you might use bacterial burden, but you might use other things like survival or histopathology or inflammatory markers or radiological measurements or bioluminescence. And some of these are experimental, clearly, but some of them are not, and they provide sometimes on occasions very useful adjuncts to more traditional primary endpoints such as the decline in bacterial burden. And then lastly, a point that, just to reiterate, using multiple strains that are geographically dispersed, that are well characterized, so you understand what they are and what mechanisms they're carrying, that are of established provenance, and also using strains with mechanisms that are likely to be encountered in clinical trials, I think is important and relevant. And that is the end of my presentation. So I will stop and thank you very much for your attention, and be, of course, happy to take any questions that emerge.
Thank you, William, for this informative introduction to this important but complex field of PKPD. We have heard how essential PKPD is for drug development, and I'm sure that the presentation provoked many interesting questions. And we will now proceed with the Q&A part of this session. As you can see in this slide, please feel free to submit your questions via the question window in your webinar control panel if you haven't done it so far. We have already received a number of questions. And the first question I would like to discuss is basic concepts of PKPD need a pharmacodynamic input. That's usually the MIC. What can be done if MIC cannot be determined, for instance, in case of antivirulence approaches?
So yes, the MIC is a traditional measure of the potency of a drug for its target. It is possible, of course, to construct PK and PD relationships without MICs, although it becomes difficult to provide a theory of knitting strains together, of understanding the variance between strains. There are potentially other in vitro tests that could, one of the reasons that the MIC is so important is because it's standardized and we all know how to do it. But that doesn't, of course, mean that you can't use other measures of potency if you could develop them. That may require some developmental work, or there may have to be some sort of scientific endeavor or research done, but there are other measures that can be used. You could use EC50s, you can use MBCs, minimum bactericidal concentrations, you could use MECs, you can use, maybe there are other readouts from in vitro assays that could be used. So I guess that the value of the MIC is in terms of understanding variance, the difference between the response of one strain versus another. But that doesn't mean to say it's the only test that could be used. It's the traditional test, but it doesn't mean it's the only test, and it shouldn't halt pharmacodynamic evaluations, and it certainly isn't an obstacle for using these techniques.
Yeah, a complex topic too. I have another question for you, William. How do you find the balance between wild type and resistant strains when developing an in vitro PKPD package?
So we would often start with wild types, which are relatively well characterized organisms that people would expect to see, but of course that may not test what you're really trying to do with the drug, and that is to demonstrate its efficacy in terms of drug resistant strains. Well, I think that it's important to achieve some balance in the program, so that there are relatively equal numbers of wild type and then resistant strains. And the resistant strains may be not only grouped in terms of the MICs, but also resistant mechanisms of importance. Then there's often a practical problem here too about making resistant pathogens grow in vivo. So some of them may not be that virulent, or their pharmacodynamics can be altered by the presence of a resistance mechanism. They actually may be paradoxically somewhat easier to treat, even after the MIC is corrected. So it may be that quite a large number of resistant strains have to be examined. But in principle, I think it's important to have a balanced design. So you don't do 8 wild type and then 1 resistance strain, or the other way around.
Yeah, another question that may be related. It's about demonstration of lack of cross resistance. What studies? What studies would you recommend if the MIC of a new derivative of a known class, so a known class, is not the same as the wild type, but still below the expected breakpoint? So just elevated MICs. Is this already demonstrations of the lack of cross resistance?
I think that it's a necessary first step to show that in vitro with MIC testing, but especially if you're going to claim that your new drug is active or is naive to the presence of those resistance mechanisms, even if they're contained within the wild type, and you're going to plan to enroll those patients in clinical trials, that demonstrating explicitly that the pharmacodynamics are not affected is an important step. So an example might be showing that a new carbapenem, for example, may still retain activity against ESBL producing gram negatives, so that the ESBL producing gram negatives may reside in the wild type for the new carbapenem, but showing that the pharmacodynamics is the same with the ESBL containing organisms versus those that are clearly wild type for all drugs is an important step to take.
Yeah, moving on to the next question. Those fractionation studies are really complex, as you mentioned already in your talk. Could you please explain again what is dose fractionation and the units used for the parameters?
Yes, so dose fractionation tests the idea about the impact of the schedule of drug administration. That's the relevance of whether a drug is given more or less frequently is the simplest way to put it. Experimentally, this is most efficiently achieved using isodose experiments. So the same total daily dose is given, but that same total daily dose is given in different schedules of administration. So some of the schedules will be more frequent and some will be less frequent, but the same total amount of drug is delivered to the hollow fiber or to the animal or even the person. So then really it's as simple as this, that if more fractionated regimens do better, so that if you give the drug more frequently, if the outcome of that is that there's a better outcome, there's more bacterial killing or there's more survival, then the effect is time dependent. If the less fractionated regimens do well, then the peak to MIC is generally the index that is important. And if the effect is the same regardless of the schedule of administration, then those drugs AUC to MIC driven. Now there are lots of nuances, but that's the general idea. As for the units, these measures are unitless. So we don't really talk about units like we do, of course, for peak concentration on its own or AUC on its time or time on its own. But the parameters or the indices themselves we don't talk about them having units.
Yeah, thank you. Another question that's also related to dose fractionation. Can you discuss in vitro versus in vivo dose fractionation for combination therapies, please?
Wow, okay, that's a nice easy question. The way that I've approached this in the past, and this is a pro forma for all combination studies, is that the pharmacodynamics is generally determined by the two drugs in isolation as monotherapy. So you do drug 1 on its own, you do drug 2 on its own, and to identify the relevant pharmacodynamic indices for drug 1 and drug 2. And then when you study the combination, you would bring them into a combination with their optimized PD. So you'd bring them both in with their relevant pharmacodynamic index, and the experiment would be set up in a way that both of the indices were optimized. So if it was a time drug, for example, you might design that so that you'd be using quite fractionated schedules of administration, and you would quantify the effect in terms of the relevant pharmacodynamic indices. But of course, the assumption that would be made in that sort of design and approach is that the pharmacodynamics wouldn't change in the presence of another drug. So it would be the same with the drug in monotherapy as it would be in combination.
Yeah, moving to the next big question. How do you need to modify your PKPD package to account for the target indication? Should you focus on strains, on drug levels, or on something else?
So by that I mean, I assume the question means the target indication being the target disease. So for example, pneumonia. Well, I hope that that's the question, and maybe it could be clarified if not, but I think that there are important differences in how a package might be designed for a disease like pneumonia, which is a good example. So that will require generally some more specialized testing. So it might require a laboratory animal model of pneumonia rather than a thigh model, for example. It would require a hollow fiber model where you actually knew what the concentration of drug in not only the blood but also the epithelial lining fluid was, and that would be put into the hollow fiber using an inoculum that would be found in those patients, so maybe 10 to the 8th per mil. So I think that there are times when the indication does matter, and it all comes back to this idea of, well, the biological relevance of the work that we do, so that there are times when it becomes very important to reproduce the disease in the systems that we're using, in the experimental systems that we're using. So I think the time has passed when you could develop a drug for pneumonia, for example, but then rely simply on a thigh model or simply on a hollow fiber model. And maybe the daptomycin experience teaches us that point explicitly. So it does become important for some drug pathogen combinations to reproduce or simulate or recapitulate the human biology and make the case explicitly. But it's very hard to write that down, I think, and that does require a degree of debate, coming back to the relationships that have to be present to make all of this work.
Let's come back to the hollow fiber studies. Would you do hollow fibers experiments only after phase 1 data are available, Or when would you advise to start hollow fiber studies?
Yeah, so this is a perpetual problem in that, so first of all, hollow fiber experiments are exceedingly expensive to run. So it's not a time to be guessing about experimental design or sort of guessing your way through. So the real problem with hollow fiber is that you're in control of the PK. So you have to decide which PK to put into a hollow fiber. And of course, one of the problems is that you may not know that early on because you haven't done any human studies yet. So in that case, it may be reasonable to use some allometric scaling to try and figure out what a human regimen or a human concentration time profile might look like. And it may be that you've got to go back and refine the hollow fiber studies once you get the real data back, in case there are any major discrepancies. But it also, even without those data, it may be worth considering some early hollow fiber work for several reasons. One is to de-risk the study by the time that you get the data. So it may be that your compound binds to the circuitry and you just can't use hollow fiber. That might be one reason, and you could find out find that out relatively early. And the other nice thing that you get from hollow fiber is getting a handle on how much drug resistance may be. So it really enables you to model the emergence of drug resistance, which you can't do in lab animal models. And you may want to see that relatively early. So in reality, I think it's very hard to put hollow fiber in these developmental pathways in a pre-specified place. It may be that you have to do some work, get some more information elsewhere, and come back into hollow fiber models. So they're iterative. So a good example might be, again, developing a drug for pneumonia where you look quickly and early in hollow fiber with some plasma PK, but you may have to come back and do those studies again once you've got some information about penetration of drug into epithelial lining fluids. So there are loops, I think, is the best way to answer that question. And again, it's very difficult to say definitively where those studies should be placed.
Yeah, it would be easy to have recipes for PKPD, but unfortunately, that's not possible. I received another question. Do we need stronger or even different PD targets to avoid resistance for the new compounds? Are efficacy targets enough?
Well, so so almost invariably resistance endpoints are higher than efficacy targets, almost invariably. And the simple answer to the question is, of course, it would be fantastic to take a very rigorous target. So maybe not only orders of logarithmic killing, but also targets that completely prevent the emergence of drug resistance. Well, it comes back to that triple lock slide that I showed, where if you want to have a very rigorous target like that, then either you can't get across the wild type, or you have to generate such huge drug concentrations that you don't have toxicity coverage. You have to use exposures that are beyond what's been established in GLP. So then it becomes it's a trade-off between wanting to take as much as you can, but still being able to develop the drug. It may be, I guess, that you may decide after doing those studies that the risk is too great and that you'll lose the drug very quickly to the emergence of resistance. And it may be that you can use these studies to take the drug down a different pathway if the risk was too great for you. But so everyone would like to have endpoints that prevented the emergence of resistance, it's just sort of very seldom feasible to do that. Of course, it does bring up the whole question about combination chemotherapy. That I might suggest is a topic for another webinar, because not only is that sort of technically difficult preclinically, but of course it's from a regulatory perspective, it's also difficult. But maybe we're going to have to do more and more of that in the future to try and address that very question.
Yeah, some new antibiotics are highly protein bound. How do you account for high protein binding in PKPD?
This is true that some compounds are highly protein bound. So the issue with protein binding is you really start struggling with it as a concept if there's discrepancies between mice or other laboratory animal models and humans. That's when the very high protein binding can be problematic. My only comment, Ursula, is that estimates of protein binding are notoriously variable and dependent on method and maybe dependent on laboratory, and that in that circumstance, because relatively small changes in protein binding can have such a profound effect on the way the data are interpreted, that it's worth spending time and money being as clear as you possibly can in terms of getting a credible estimate. Of course, as soon as you've got that estimate, then someone will say, well, what about protein binding in a critically ill patient? Or what about protein binding across a range of concentrations that I could expect in patients? And they are all valid criticisms that will have to be dealt with. And then the other thing to say is with hollow fiber is that generally, well, almost invariably, those studies are done without protein. So that there's in that system, protein binding is taken out of the equation, as it were. And so that has the effect, or the pharmacodynamics in those systems has to be accounted for in that situation. So I will just sort of summarize by saying I wish I really did know the answer to that question. Sometimes it can be very challenging to deal with, especially, like I say, if there are discrepancies between laboratory animal models and humans.
Yes, we have a lot of challenging questions here. So the next one, can the same PKPD approach be applied for antimicrobial peptides?
So potentially, yes. I think the challenge there is just being clear about handling the PK. That's generally the challenge that I've found is how the PK are handled, whether you can measure the drug and whether you can quantify drug exposure in those more traditional ways that are available or have been developed for small molecules. But conceptually, the answer for small peptides is yes. And of course, assuming that the peptide induces some sort of traditional antibacterial effect rather than it modifies immune effectors or has an effect on virulence. But if the question was that it docks with and kills bacteria, then yes, with a proviso that you can measure the peptide.
Yeah. Based on your experience from the clinical point of view, 2 log drop and 90% probability of target attainment versus 1 log drop, 99% PTA, which is more appropriate?
Right, so this is the very question that I worry about, because remember that these are all absolutes. So 2 log drop, 1 log drop, 90% PTA, 99% PTA. So I think that there are 2 answers to the question. So the first is if you were developing this drug for pneumonia, you would take a 2 log drop and a 90% PTA. If you were developing the drug for a less severe indication, then the latter may be appropriate or relevant. But increasingly, our own response to this problem is to think about the relevant comparator that's been planned for the clinical study, and then try and establish the pharmacodynamics of that compound in the same model systems, so that let's just say that the comparator was levofloxacin. It may be that the performance of levofloxacin in the same model systems would be important to establish using the drug exposures and the dosages that are going to be used and planned, and demonstrating that, or being explicit about how much effect that comparator induces, and planning to match or exceed that. And that's this concept of benchmarking. So that's my answer to that.
Yeah, I have a related question here. Is there any regulatory guidance or other guidelines on the probability of target attainment that is required for drug approval? Is 80% adequate?
No, I think in the EMA document, we just need to go back and check this, but they talk about 90% at a given MIC value. Now, you know, again, this is this problem of being too rigid. But that often is again, whether you say it's 80% or 90%, there's this concept again of trading. It's this triple lock problem of trying to get across the wild type, and that in turn depends on the dosage that's used, and that in turn depends on how much, coming back to the resistance endpoints and stasis and 1 and 2 logs, that you get into a bind between these various things or parts of the problem that you're trying to solve. So it's very difficult to be, it's very dangerous to be too absolute or too inflexible about that. But I think the EMA talk about 90%, but just that document's in the public domain and just worth checking.
Okay, thank you. How can a PKPD index be expressed for a beta-lactamase inhibitor? Is it possible to use the MIC of the combination, for instance, time above MIC of the combination?
So the traditional, the best approach to this is unclear. And there are a number of different approaches that have been used. So of course, BLIs tend not to have any intrinsic activity themselves, and so you can't establish therefore an MIC. So the MIC, or maybe to say it more precisely, a threshold level, has to be inferred from experimental data. So the MIC is not determined in an in vitro test, but it's determined from setting up an experiment. And there are a number of different ways that this can be done. So one way is that, so rather than talking about MIC, it's perhaps better to talk about threshold levels, which, and I'm sorry to say this, they may not have any biological or intrinsic relevance, but they're a way that you can use to describe and talk about the data. So there are two main ways that this is done. One is to use regression analyses to try and figure out what that threshold level may be. So different thresholds are tried in the regression, and which number gives the best fit is brought forwards. Or another way that this is done is it can be done in a hollow fiber model where a drug is infused and at a certain level, and then the pharmacodynamics are observed. And so those concentrations or thresholds in a hollow fiber model that enable or result in bacterial killing and prevent the emergence of resistance can be then brought forward as a descriptor. So rather than the MIC, it's a number, and it's probably better referred to as a threshold, and it's just a way that the data can be handled and described rather than necessarily having intrinsic meaning.
I have a related question here. You mentioned spiral therapeutics. They are following an adjuvant therapy approach, with one of the two molecules having very little or no activity per se. Would you do any dose response studies with both compounds before deciding on the dose for speculation studies?
Yeah, so this is sort of analogous to the beta-lactam BLI problem, where one of the compounds doesn't have any intrinsic activity. And so if I can just talk about the beta-lactam beta-lactamase inhibitor design, then well, our own approach is to examine and fix one of the compounds, and usually that's the backbone. So it's the beta-lactam where the regimen is fixed, and then the second compound is then studied in terms of different dosages and schedules to discover or uncover the pharmacodynamics. But the same problem about what you use as the descriptor for potency arises in that situation. So that this is an analogous problem, and maybe it's going to become increasingly the case using compounds that we know and can see are biologically active, but we can't measure their effect in a test tube and generate numbers like the MIC.
Yeah, we are discussing the MIC concept in the PKPD community for a long time. And I have another question to MIC. MIC evaluation is conducted under equilibrium in vitro. However, in vivo drug and target are not at equilibrium all the time. How MIC value can be used as a good metric for PKPD evaluation in vivo system?
Yeah, so the MIC is nothing more than an in vitro test that occurs under a set and agreed set of experimental conditions and yields a number that has necessarily no intrinsic or absolute meaning as a value, and its only purpose is to explain variance, and so it's a measure of the potency of the drug for its target, and nothing more and nothing less. And the corollary of that is that an organism that has double the MIC requires double the amount of drug exposure to produce the same effect. So I think it's just important. It's a scalar is another way to say it. Its intrinsic value and the way that it's determined doesn't necessarily have any intrinsic biological relevance or value. It's just a way, again, of explaining variance in the data and predicting what might happen in the future. But other than that, I agree that it's that it is conducted under equilibrium in highly sanitized conditions. I agree with all of that. And those people that do MICs know how difficult and how infuriating they can be to do, and so sort of also how inaccurate they can be, given that we use doubling dilutions often.
Yeah, considering the use of stasis 1 log or 2 log drop endpoints, how does using an immune competent animal versus neutropenic animal change consideration of the appropriate endpoint to choose?
Yeah, I think that's an interesting question, because many of us don't use, well, we tend to use neutropenic models of the mouse, and for sure it's possible to use immunocompetent models, and they're trickier to use because immune effects are non-linear. They happen, and when the immune system turns on and starts, there's clearing, immunological clearing, that's usually highly effective. So these model systems are quite unstable, and it can be very difficult to distinguish what might be immune effects versus what might be drug effects, even with properly controlled experiments. But I think that without having faced this, well, I've done immunocompetent models, but never resorted to using stasis 1 log or 2 log endpoints, and I don't think that is necessarily appropriate, because their meaning is all being derived in the context of neutropenic animals. So again, if I was asked to interpret logarithmic drops or orders of logarithmic killing in immunocompetent animals, I would be very clear to use a benchmarked example, I think. So I could compare my new drug with the way that a standard comparator was behaving. But I don't believe that we should use stasis in 1 or 2 logs in the same way as we would for a neutropenic animal or background.
We didn't talk yet about topical antibiotics. How would you study the PK of a topical antibiotic? That's a difficult question.
Yeah. Well, I'm going to answer in a very, very, maybe in a difficult way. I think that, this is an unexplored field. I will say I think that there's interest in, and I think that there probably has to be a whole new science invented for this problem and problems like this, and that the PK now depends not only on time, but it depends on space. And there is a discipline called spatial PK, which is sort of moderately, if not extremely complex, because you actually have to solve in two domains, space and time. But it probably is relevant to many of the processes that we study, such as, well, for sure, topical therapy, but then also one might consider the way that drugs penetrate into an abscess, or the way drugs might penetrate into an endocardial lesion, a valvular lesion. So spatial PK is probably a lot more important, but we, of course, assume that the drug has instantaneous access to organisms wherever they are in the body, and that's probably not true in the majority of cases. So I can't really answer the topical PK question in a more precise or refined manner than that, other than it needs a whole different approach to the paradigm that I sort of outlined in the talk.
I'm coming back now to an overall clinical question. In clinical trials, there's some discussion about the difficulty or even inappropriateness to perform superiority studies, while non-inferiority studies have pitfalls. Can PKPD be used to bridge this gap with regard to regulatory demands?
I think that might be just in 2018, maybe a bridge too far for PKPD. I think that it is possible, though, to do a better job than we currently do of setting out the pharmacodynamics of the new drug versus the pharmacodynamics of the comparator. So generally what will happen is that a comparator, the pharmacodynamics of the comparator will be assumed, actually. They'll, so there might be, you might hear a comment like, well, penicillin has to be 50% time above MIC to be effective, rather than demonstrating that explicitly in studies parallel to the new drug. So in terms of the question really strikes to the heart of drug A versus drug B in terms of non-inferiority or superiority, but it may be possible to set up the experiments in a much more head-to-head way to at least de-risk the clinical study in whatever the design is, by de-risking the pharmacodynamics, so making sure that drug A is set up favorably in comparison to the dose that's going to be chosen for the comparator. But I'm not sure whether PK and PD actually would help necessarily be a bridge to the problems of superiority or the gamble of superiority, but it can for sure, I think, do a better job of de-risking that problem, so at least the appropriate choices can be made in terms of design. I'll have to think about that some more. It's a good question.
Does the PKPD approach could be used to study phage-based therapies? What are the main challenges? Again, a very big question.
Yeah. Well, I think in principle, yes, but I think this question starts to get at some of the challenges that lie ahead. So what I described was very much a well-trodden pathway. Well, that's, even though it's developing rapidly, it's sort of, as the EMA says, is mature and fit for purpose. But the paradigm is very much around small molecules and around sort of standard bacterial pathogens, as it were. Now, as soon as you deviate from that, you're going to have to invent some new science, and of course, that immediately is going to make everybody slightly more nervous if they're the first one to pursue a path. So then I can only say that this is something that we all have to do. That it's exciting to do, but it just requires a whole lot of things. So it will require robust validation, it requires benchmarking. It requires early discussion, not only with the scientific community, but also the regulatory community, to try and find ways through this. But in principle, I don't think that that the phage problem can be is amenable to some of the approaches that I've described, so that you have in vitro measures of potency with in vitro testing. You know how much of the phage that's put in you. I guess you have an estimate about how much it expands, and you can observe the decline in bacterial burden. Well, I just think that I'm not pretending that that's an easy problem, but I think that, again, as for virulence modifiers, these are problems that we're going to have to solve, and we have to find new science, and we have to convince ourselves and others that the models that we make and the approach is relevant, and that only comes from good science and good data.
Scientific community will be kept busy in the future. I have still a lot of questions. One of them is, are there any attempts to incorporate or include inter-individual variability of host immune responses when doing this type of studies?
Well, rudimentary is the answer to that. So the best work I know of comes from George Drusano, who incorporated neutrophils as adjuncts into as what enabled the neutrophils to have their own antibacterial or antifungal effects, and that was in addition to the drug-induced effect. And so it's possible to use the same PKPD or the mathematical modeling techniques to build in estimates of immune effectors. And neutrophils are relatively easy to do because you can count them, and you can count them as frequently as you look. I guess there's absolutely no reason that more other metrics of immune function can't be built into these quantitative models. You just have to be sure that they mean something, that they're, in other words, they're influencing the outcome, that they can be measured, and that you can understand not only the variant structure, but you can also understand how the immune system is having an impact upon the ultimate antibacterial effect. So yes, for sure this should be done because it's clearly relevant to patients. Of course, in experimental systems of hollow fiber, there are no immune effectors, and in mice, we do everything we can to remove those effects because they confound results. They're very difficult to control. So the experiments are difficult because the immune system, when they come, is so much more powerful in general than the drug. So you're never quite, and the effects are non-linear. So again, like the phage question or the antivirulence drugs, I think that this is that just much more needs to be done on this in the future. And yes, it should be sort of incorporated into models. So I'm just cautioning that it's not straightforward.
The next question is related to an earlier question regarding indications. How important is it to conduct lung penetration studies, considering the inconvenience to patients? Is there any acceptance of utilizing PKPD approaches to estimate lung uptake and distribution as an alternative to a clinical study?
Well, I think especially the European regulators are pretty adamant that in terms of developing a drug for pneumonia, that they would require at a bare minimum that there was a lung penetration study in healthy volunteers to estimate the degree of penetration of drug into the epithelial lining fluid of volunteers. They generally don't require patient studies, which are of course notoriously difficult to do because patients are ventilated with all the attendant issues or problems. I think this again is a difficult area. I think that maybe the range of opinion goes from just being satisfied that there's some drug in the lung and that being at least something to de-risk a future clinical study in pneumonia, versus a much more rigorous interpretation of the data where the amount of drug that's in epithelial lining fluid actually is responsible for driving the antibacterial effect, and it's possible to make the bridge from preclinical models through healthy volunteers to patients based on the amount of drug that's in epithelial lining fluid. And well, there are some examples where that appears to be the case. There's the keftabiprole example as one that springs to mind. So I think that the standard approach would be to at least do lung penetration studies in volunteers and quantify that, quantify the degree of lung penetration as much as possible. But I think that there is some uncertainty about how to process the data, or how literally to take the data, whether one just proceeds on the basis of other measures of dosage identification, or whether to actually drive the whole program of the concentrations that are achieved in volunteers. There's also some emerging data now that lung penetration for at least one or two drugs is different in patients compared with volunteers, and I guess that shouldn't surprise us. So the point estimates of penetration are different, as well as the variance structure. So this is, yeah, it's a notoriously difficult area again, but at a bare minimum, European regulators for sure will ask for some lung penetration studies in volunteers for a pneumonia indication.
Yeah, you just mentioned the differences between volunteers and patients. How essential is it to measure ELF concentration or other tissue site PK in critically ill patients, in addition to healthy volunteers and patients in normal wards?
Yeah, and again, the question is, if I heard it right, it was about is about really the critically ill setting and whether lung penetration may be different in patients. And I think the assumption is that it probably is going to be different, not only in terms of more variability, but actually the penetration may go up and it may go down. It may go up because of there's hemorrhagic infarction or there's inflammation, or it may go down because there's interruption of active transport mechanisms. So it's very difficult, I think, to be sure about which way it's going to go. Well, that has to be balanced against the considerable difficulty of getting lung concentrations from patients, not only because of the consent issues, but also the potential risk of going into a critically ill patient with a bronchoscope. I suppose you can do these studies with relatively small lavage volumes, unlike in volunteers, which tend to be much larger. But nevertheless, it's a difficult area. And then, of course, another problem that you've got is that how many patients should you enroll in those studies, because each patient's only going to realistically yield a single observation, so that the data are generally may not be optimally collected. That may occur anywhere in the dosing interval. So for sure, I think that making the case for studying patients is, it may be just a bridge too far in terms of doing the study and then being able to reliably interpret the data. I think that it's pretty reasonable to me to make the case to study volunteers and recognize that it's imperfect. And of course, you can also study laboratory animals pretty easy and see how the two compare. But the patient study I think is a more difficult problem, and I think that you have to be very clear about what you're hoping to get out of a study like that to justify the cost and the intervention.
Thank you. I'm moving on to the next question. How often does the initial in vivo animal PKPD predict accurately the human PKPD? How much iteration is normally required?
The human PKPD. So well, but it depends a little bit on the drug pathogen combination. So remember, I guess, just to break that question down a little bit, that it's generally, of course, not possible to do dose fractionation studies in humans, and so one relies on the variability that you see in humans to see whether there are different different impacts of various PK and PD indices on outcome. So and generally there's just not enough variability to be able to break the covariance and be able to say one is better than the other. So it's very hard. So it's almost invariably the case that the relevant index is established in preclinical models and assumed to be the same for humans. However, there's generally pretty good concordance between the magnitude of the index, however that is quantified between preclinical models and humans. Of course, that's dependent on the context. So it may be that more drugs needed for a critically ill patient compared with a less unwell patient. But the degree of concordance, of course, allows us to make statements that were in the earlier questions about the relevance of stasis and 1 logs and 2 logs because of that historical body of experience where those links have been made. So I hope that answers the question. So for fractionation or the determination of the index is not possible, but the magnitude of the index, once it's determined, seems to correlate pretty well. But of course, there's just one other thing to say, and that is that the whole purpose of doing PK and PD is so that you try and minimize the chance of seeing those relationships. In other words, you try and deliver a dose to patients that maximizes their chance of recovery or clinical benefit. And so that if you see sort of significant PK and PD relationships in patients, especially in a phase 3 study, then sort of something pretty bad has happened where you haven't anticipated what you might see. And I guess so that's a situation that in general you want to try and avoid if at all possible.
Yeah, unfortunately, we have run out of time now. It was a very interesting discussion and great questions. And thank you very much, William, for taking the time to really try to answer some of these difficult questions. For those of you who have posted additional questions, we will put them on the public forum of the Revive website in the next few days, and we will do our best to respond to these questions. So thank you very much again, William, for this discussion.
And here is Francois again. So make sure to visit the revive.gardp.org. You can find there recordings of previous webinars and conference sessions, or you will also find there announcements of future webinars, as well as the public forum where we will post the questions that we didn't have the time to answer because we had a couple of hundred attendees. And importantly, also, you will find there a section called Talk to an Expert. This feature allows you to submit questions to us, which we will assign then to specific experts. And once we find the right expert for your question, you will be able to discuss further with that expert in a confidential manner outside of the Revive website. With this feature, we want to help researchers who are not yet well connected in this field to gain access to world-renowned experts that have volunteered to be part of this pool at GARDP. And with this, I would like to thank William and Ursula for this webinar. And I would also like to thank everybody for joining and for contributing to the discussion. Thank you so much.